Hello everyone, and welcome to this audio podcast in this series, the LabZtorm effect v.3. This is an audio podcast aligned to the role of endogenous opioids in ischaemic preconditioning, or IPC in humans. This is an audio podcast that looks at and that will look at and sort of delve into endogenous opioid peptides, or EOPs, in RIPC, or remote ischemic preconditioning. This is an audio podcast aligned to a storm robotics soliloquy and a storm robotics another storm robotics production. Um, and um, and so So in and around endogenous opioid peptides, or EOPs, in remote ischemic preconditioning. Um, so before delving into this, I want to before delving into it and delving into this specifically, or particularly, um, to highlight some aspects in around the sort of pharmacological basis in around cardioprotection. Now, search for pharmacotherapy strategy to protect the heart against ischemic reperfusion injury, or RI. Oh, RIR, or IRI, preceded the discovery of IPC, ischemic preconditioning, by many years. The elucidation of signaling pathways underlying ischemic conditioning have resulted in advances in the understanding of pathophysiological mechanisms of ischemic reperfusion injury, and have identified a number of molecular targets amenable to pharmacotherapy manipulation. The history of pharmacological cardioprotection has at the same time been somewhat at the same time disenchanting. So anti-inflammatory agents, antioxidants, calcium channel blockers, and other and pharmacotherapies, including erythropoietin, for example, and magnesium, have all been ineffective in reducing the size of MIs, or myocardial infarcts, and improving clinical outcomes. A number of pharmacological cardioprotective, or CP strategies have cardioprotective strategies, such as, for example, adenosine, glucose related treatments or therapies have showed mixed results with CP or cardioprotective efficacy, depending on the study design. Now, in around this and in around this and sort of particularly on EOPs, or endogenous opioid peptides, in RIPC, originally termed opioid, was coined by Aitchison to place drugs whose mechanisms resemble, for example, a morphine, but chemically distinct from its structure. It was in 1954 Beckett and Casey who hypothesized that synthetic analgesic opioid drugs and related morphisms sort of fit at a receptor surface, stereospecifically, and it was through sort of interaction at the surface, at a receptor surface, that analgesia, or pain relief actually had been identified or established from works by Beckett, by works in the 1950s in the 1950s by Beckett and Casey, along with others. And it was not until the sort of 1970s, with the advent of the ligand binding studies, for example, that the agonist opioid receptor relationship or interaction or interplay could be sort of exemplified or probed. And sort of more prospectively, data has suggested that opioid receptors are important triggers and or mediators of cardioprotection or a response. Opioid receptors, for example, are known to be G protein coupled receptors, or GPCRs, whose activation inhibits adenyl typical of many cardioprotective GPCRs. So, for example, adenosine and bradykinin. Accumulating evidence support the notion of an intrinsic opioid receptor system in and around cardiovascular or cardiac and around the cardiac muscle, and these contribute to functional changes in normal and deceased myocardium. It was Hughes in the 1970s, in 1975 specifically, who showed EOPs, endogenous opioid peptides, that could be isolated from brain extracts, and that these peptides posed morphine-like properties. Now, works by Martin and co-workers in the 1960s and 1967, specifically using congeners of morphine, obtained the first in vivo evidence of opioid receptors. The investigators had identified three distinct syndromes produced by these agents and the use opioid receptors. The investigators identified three distinct syndromes produced by these agents in canines, and identified them as specifically as opioid receptors. In principle, there are typically three classes of opioid receptors. There's the opioid receptors aligned to mu, and there's opioid receptors that are aligned to kappa. And then there's opioid receptors that are aligned to delta. It was works by Lord and team in 1977, wherein the team has implemented works using the mouse vas deferens, endorphins, and enkephalins as agonists that brought imperative distinction and confirmed the third opioid receptor, known as the delta receptor. Now, the sigma receptor is no longer considered, as it was as once before, to be an opioid in nature. Its physical and pharmacological actions are not blocked by opioid antagonists, for example, Naloxone. When the sigma receptor was cloned, it was identified to have no structural similarity to opioid receptors, and it was at this point that they were designated as a separate class of receptors, and the function of sigma receptors has been generally poorly poorly documented and sort of understood. It was worked by Schultz and team in '95 who demonstrated that opioids involved preconditioning and some aspects of post-conditioning using in vivo models, and it has been suggested that opioids play an intrinsic role in the mediation and regulation of around the regulation of the cardiac muscle in both normal and diseased in normal and diseased models. It was also worked by Schultz and Gross in 2001, who reported the capacity for the cardiac muscle to synthesize all three types of opioid peptides aligned to enkephalins, endorphins, and dynorphins. It was worked by Pugsley in 2002, who intricately discuss it in their work in research and documented works and research the molecular biology of opioid receptors. The endogenous opioid peptides EOP system consists of peptides endorphin, dynorphin, and enkephalin, and associated receptors. The opioid peptides and receptors are widely distributed in context of pathophysiology, and all have complex actions in around the cardiac muscle. Endogenous opioid peptide receptors or EOP receptors derived from hormone precursors as well from hormone precursors, and EOPs are distributed throughout cardiac nerves, intrinsic nerves, and gastrointestinal tract, and again in around the cardiac muscle. Cardiac opioid peptide function is complicated by the fact that the receptor expression is modulated by both changes in physiological and disease sort of states. Now, it's works by Pearl and team in 2005, who reported that chronic opioid exposure leads to a phenotype consisting of profound ischemic tolerance state, and the action of opioids are mediated by opioid receptors located centrally in specific areas of the brain and the nuclei that regulate the control of cardiovascular function, and preferably by tissue opioid receptors. Whilst no overall consensus has been sort of retrospectively been established regarding the role of opioids and of the role of opioids in context of interplay and regulation of the cardiovascular system. They have been implicated in several significant cardiovascular disease conditions, or in around CVD or cardiovascular disease, and whether endogenous opioid peptides or EOP titers increase or decrease during ischemic-reperfusion injury, or whether significant titer changes occur during ischemic-reperfusion injury is still has still been of some intrigue in literature. Several investigators have found that cardiac ischemic-reperfusion injury result in an increase or decrease of plasma opioid peptide level, whilst others have found that there's been no change or decrease in EOP activity following ischemic-reperfusion injury. In turn, the effects of IPC and opioid agonists to produce cardioprotection are most likely the effect of delta or kappa sort of opioid stimulation. In animals, evidence supports delta opioid receptor as preliminary responsible for mediatory for the mediary role in ischemic preconditioning, and binding studies reveal the presence of kappa and delta receptors on ventricular myocytes in laboratory rat models. Endogenous opioid peptide distribution, or EOP distribution has been highly sort of variable within cardiovascular parenchyma. At the same time, retrospective animal investigations, laboratory animal investigations have supported the notion that delta opioid receptors and potassium adenine triphosphate, or potassium adenine triphosphate channels, or KATP channels, mediate cardioprotection, or CP effects of ischemic preconditioning, or IPC, in rat laboratory cardiovascular models. The physiological significance of opioids in protection or cardioprotection against ischemic-reperfusion injury in humans has still not been explored intricately. Has not been explored sort of intricately, and is not fully understood. In this general in this regard, or in this on this basis, sort of mu opioid in this regard, and in regard to this, the mu opioid Beta-Endorphin and Beta-Endorphin has warranted attention and sort of critical has warranted attention and critical appraisal. So this ends the this particular audio podcast in around endogenous opioid peptides, or EOPs, in remote ischemic preconditioning, and in line to this audio podcast, the role of endogenous opioids in ischemic preconditioning, or IPC, in humans. The next audio podcast in this particular series around the LabZtorm effect v.3, we'll look at more specifically the Beta-Endorphin and sort of the audio podcast in around Beta-Endorphin. We'll delve into some of the investigations, and some context in around what has been sort of has been reported in this context, at least in some part where investigations have been sort of documented. So this is another audio podcast. This is another audio podcast as part of the Storm Robotics soliloquy. It's been another Storm Robotics production. Thank you for tuning in.